Good afternoon, everyone. I'm Martha Pantelidou, and it's a pleasure to be here at the FishBase and SeaLifeBase Symposium in Thessaloniki. Today, I will be presenting our lab's research on how sublittoral rocky habitats in marine protected areas of the Aegean were affected after the severe marine heat waves of 2021. Before I dive into the main topic of my presentation, I would like to briefly introduce our lab and its research focus. Our lab focuses on a broad range of marine diversity topics. Firstly, we investigate marine biodiversity with an emphasis on benthic invertebrates, addressing the threats and focusing on conservation efforts for species, habitats, and marine protected areas. Our marine ecology research includes studying benthic communities and ecological succession in a broad range in a lot of different substrates. Another key area of our work is invertebrate biology and fisheries, encompassing numerous marine organisms such as sponges, cnidarians, mollusks, crustaceans, and a lot of others. Lastly, we conduct ecological quality assessments based on benthic macroinvertebrates as benthic quality indicators in coastal and transitional waters. The present study aims to assess the status of shallow sublittoral rocky habitats in the 5 special areas of conservation of Chalkidiki, following the 2021 marine heat waves and by applying non-destructive techniques. Currently, the establishment of marine protected areas is one of the most widely applied tactics to preserve marine biodiversity. However, their resilience to global threats is doubtful, since most of the existing marine protected areas were established without considering these stressors. In view of this concern, adaptive management is required by setting and monitoring appropriate biological indicators. Adaptive management is a proactive and flexible approach that allows for the continuous improvement of management practices in response to changing environmental conditions. Among the proposed biological descriptors, various sessile epibenthic invertebrates are highly recommended due to their prominent presence on shallow sublittoral rocky shores and their sensitivity to sea warming inducing mortality events. As I mentioned, in 2021, successive marine heat waves affected the Aegean Sea, the north sector in particular, including the special areas of conservation of the Natura 2000 network. Our study area was Chalkidiki Peninsula, located in the North Aegean Sea, and we selected 14 sampling stations in 5 special areas of conservation within Chalkidiki Peninsula. 6 of these stations, as shown in the map, are in Cassandra, the first peninsula of Chalkidiki, and 8 of them in Sithonia, the second peninsula of Chalkidiki. Sampling was conducted in September and October of 2021, using scuba diving techniques in the infralittoral zone, up to 30 meters of depth. We employed vertical belt transects to assess the diversity, the relative abundance using the ACFOR scale, and the health condition of the most prominent epibenthic organisms. The ACFOR scale, ACFOR stands for abundant, common, frequent, occasional, or rare, is a semi-quantitative method to assess the relative community composition in larger areas or rugose environments. Overall, 57 macroscopic species, sedentary species, were recorded, 13 of which were algae species and 44 were invertebrates. Species richness ranged from 10 to 46 species per station, with an average of 25 species, and with more species present in deepest and steepest cliffs. The names of the species detected are given in detail in this slide, and apart from the sedentary species, fish like scombers, red mallets, groupers, and sea breams were also very abundant. According to the Habitat Directive, 2 main types of habitats were recorded during our underwater surveys: reefs, which were present in all stations apart from Station 2, and submarine structures made by leaking gas, only present in Station 2. According to the UNIS habitat typology, on reefs, the biocynosis of Mediterranean infralittoral algae occurred and the coralligenous biocynosis. The biocynosis of Mediterranean infralittoral algae exhibited phases of pavonica, Chistozeira, Halopteris coparia, and Corallina, and also colonies of Cladocoraca spitosa, whereas the coralligenous biocynosis exhibited phases of Halimeda tuna and Lithophyllum strictiforme, and also massive sponges and sparse red algae. As far as the submarine structures made by leaking gas are concerned, 3 types of habitats, according to the UNIS typology, were recorded: the biocynosis of hydrothermal oozes, the biocynosis of Mediterranean infralittoral algae, and the coralligenous biocynosis present as enclave. The biocynosis of Mediterranean infralittoral algae was defined by Chistozeira, and the coralligenous biocynosis by Halimeda tuna and Lithophyllum strictiforme. In this part of my presentation, I will share with you some photos from our underwater surveys, including the most prominent epibenthic sessile species. In the Mediterranean infralittoral algae biocynosis, very common were sponges like Chondrilla nucula, Chondrosia reniformis, Yersinia variabilis, and Aplyssina aerophoba, the solitary stony coral Balanophylla europea, and the sedentary worms of the Serpula genus. In the coralligenous biocynosis, sponges like Agella soroides, Axinella verrucosa, Sarcotragus foetidus, and Spirastrella cuncatrix were also very abundant, together with ascidians like Halocynthia papillosa and Microcosmus abatieri, the solitary stony coral Leptosamia pruvoti, and the bivalve Spondylus gederopus. Here are some photos of the second station, which was characterized by the submarine structures made by leaking gas, and the most prominent organisms like the algae Halimeda tuna, the sponge Yersinia variabilis, and the Serpula worms. Very abundant were fishes from the species Thalassoma pavo, Mulus hermuletus, and Diplodus vulgaris. Thank you. The non-metric multidimensional scaling was used to visualize the similarity and dissimilarity between the stations. In the first graph, all detected species are included, and on the second graph, only the invertebrates. As we can see in the second graph, the stress level is lower, suggesting that the structure of the data is more accurate in the second graph. The stations are divided into 2 main groups, with the exception of Station 2, which was characterized anyway by a very unique, different habitat type. The analysis of similarities showed that the significant factors affecting the grouping of these stations were the depth, the presence of cliffs or boulders, and the inclination of the seafloor. During our underwater surveys, 15 species were found in partial or full necrosis. Accordingly, they may represent appropriate climate change descriptors to assess the resilience of Mediterranean marine protected areas. On the left, you can see photos of the algae affected, like the calcifying algae Halimeda tuna and crustose calcareous algae like Lithophyllum strictiforme or Pefsonelia species. On the right, there are sponges in partial bleaching, like Aplyssina aerophoba, Chondrilla nucula, and Chondrosia reniformis. Here are some more photos of sponges in partial or full necrosis, and on the upper right photo, the dead thorny oyster Spondylus gederopus. Lastly, the fan mussel Pinna nobilis was found distracted, also the Microcosmus abatieri in full necrosis, and the corals Balanophylla europea and Cladocoraca spitosa. The stony coral Balanophylla europea was found in full necrosis, whereas the colonies of Cladocora were found both bleached, like the first photo, and in full necrosis without any living tissue, like shown on the photo on the right. Concluding, the sponges Sarcotragus foetidus and Yersinia variabilis, the corals Balanophylla europea and Cladocoraca spitosa, and the bivalve Spondylus gederopus were the most severely affected species among the surveyed special areas of conservation. Only empty or distracted cells of the fan mussel were observed. The species used to be abundant at all surveyed stations prior to the epidemic disease. Comparison with data from 20 years ago indicates a significant reduction in biodiversity with no previous signs of necrosis observed. And the affected species used to be prominent. Their monitoring, using visual sensors, may quickly and easily reveal changes in the marine ecosystems, and therefore they may represent appropriate climate change descriptors to assess the resilience of Mediterranean marine protected areas. This research was partially funded by the Natural Environment and Climate Change Agency, and I would like to extend my gratitude to Labros Mitsoudis, captain of the Poseidonia vessel, for his hospitality and invaluable assistance during our sampling efforts. Thank you a lot for your attention.